Thank you, Aubrey, to the introduction and the invitation to this really exciting conference here. And I want to just start off to remind you that the demographic change is really a global challenge that we are really facing all over the world. And our societies are getting older, and with old age, invariably, we're getting sick. And it's not only the single disorders of aging that you can see here, but the real disease of aging is multimorbidity. So that is that the likelihood of having two or more chronic diseases is really exponentially increasing after the 5th decade of life. And this is affecting a major part of our population. If we want to tackle the challenges of the age-associated disease in our ever-aging society, we need to understand the mechanisms of aging, because only when we understand the mechanisms, we can have meaningful interventions. When we want to understand any problem in biology, we need to understand it in the context of the evolutionary history that has shaped the process. And a key realization with regards to the aging biology came from August Weismann in the late 19th century, who proposed the theory of inheritance that we hold true until this day, which is that exclusively germlines and only germ cells perpetuate the genetic information throughout the generations. And our germ cells are essentially 300,000 years old as modern Homo sapiens. And so the genomes in the germline are indefinitely maintained. In contrast, because germ cells are indefinitely maintained, the soma only needs to be maintained for as long as it took in our evolutionary history to generate the offspring. And that's why after the 3rd decade of life, we are no longer maintained, we age, decay, and die. And so there's only single generation somatic maintenance. Now, the molecule that has to be constantly repaired and maintained is the nuclear DNA that encodes all information to build and function as a cell. Nonetheless, it is constantly challenged by exogenous and endogenous damage types that come in a myriad of distinct forms. And DNA cannot be replaced. It's critically dependent on DNA repair. The DNA damage, we know for a long time, can lead to genetic aberrations, such as, for example, mutations. And it is very well established that such genetic aberrations cause cancer. DNA damage is caused by cancer. But DNA damage can also block transcription and replication, elicit DNA damage signaling, and this leads to alterations in cell fate. Cells die, cells undergo senescence, stem cell compartments are deprived, and this is how DNA damage drives the aging process. The cellular function declines, the tissues become atrophic, and inflammation becomes systemic. So DNA damage is a critical driver of both cancer and aging, and DNA repair mechanisms are essential to prevent these detrimental outcomes. Here you see the consequences of when humans are born with a defect in DNA repair. On the one side, you see xeroderma pigmentosum patients with already in childhood multiple skin cancers, exemplifying the mutagenic consequences of DNA damage. On the other side, you see a boy who suffers from Cockayne syndrome, a severe progeroid syndrome, where the patients are growth retarded and develop the same age-associated diseases that we are getting when we are very old. This boy showing retinal degeneration and died with neurodegeneration and atherosclerosis, typical disorders of the elderly. Now, can we reverse this? Can we do the opposite? We know very well repair defects, accelerate aging. Can we actually increase the DNA repair capacities? This has been hampered by the complex biology. There are hundreds of distinct damage types forms. There are at least 6 major and many additional DNA repair systems that remove these distinct types of DNA damage. And thus far, there were only very highly lesion-specific single repair enzyme reactions, such as photolysis, that could be improved. But they are blunt towards all these complexity of different damage types. We ask the questions whether there are master regulators of DNA repair existing at all. And this was driven by this observation that the soma and the germline have very different repair capacities, where the germline has far higher repair capacities. Germline mutation rates are of more of an order of magnitude lower than the somatic mutation rates. Of course, in humans, this is complex. We turn, therefore, to C. elegans, which has the exact same repair mechanisms that we have as humans, and a very experimentally accessible distinction between the post-mitotic soma and the highly dynamic germline. Now, Arturo Bujabal-Dueso in the lab took this challenge and asked whether there could be a master regulator by very simply looking at all the DNA damage response genes in C. elegans and asked whether there was any promoter sites enriched. And he found a very specific promoter motif that was the DREAM target sites, called the CDE-CHR motif. This motif is bound by a transcription repressor that's called the DREAM complex for dimerization partner RB-like protein C2S-MFP core. And this is a repressor, and because it is upstream of the repair genes, we hypothesized that this repressor would limit the expression of DNA repair genes. We knew for a long time the DREAM complex in C. elegans is only expressed in somatic tail types. The prediction would be that the worms become resistant to DNA damage. This is indeed the case. We use here this L1 early larval stage, all mostly post-mitotic cells. They grow in size through an adult worm. And after DNA damage, their growth delayed. You see it in the wild type. After UVB, there's severe larval arrest that is alleviated by mutations in distinct components of the DREAM complex. This is a bona fide function of DREAM complex. Genetic epistasis, a double mutant, is as resistant to DNA damage as the respective signal mutants and significantly resistant compared to the wild type that is arrested. We then ask whether this works also in adulthood. There's an important distinction between the simple C. elegans with a short life and humans. In humans, one repair defect accelerates aging. In worms, we need to actually provide them DNA damage. And the solid lines here, DNA damage-driven aging, is delayed in each one of these mutations in distinct components of DREAM. And do they repair better? The big advantage of working experimentally with UV is that we can quantify the UV-induced lesions, 80% are thymidine dimers. We can measure them with an antibody, and we see here in the slot plot that there's inefficient repair in the wild type, but much more pronounced repair here in Lin52DPL1 in the DREAM mutants. The higher expression of DNA repair genes leads to better repair kinetics. It's also in adults. Here you see a head of an adult. Most of these cells are neurons, in fact, irreplaceable neurons that very inefficiently repair the DNA lesions that we stain here. A DREAM mutant is far more efficient in repairing the lesions. Now we ask, is this more of a general mechanism? We did RNA-seq, and we see indeed in a Lin52 DREAM mutant compared to wild type, we see the induction of many genes because of the repressor that we inactivate. And many of those are DNA repair genes. And what is unprecedented is that these are genes in nucleotide excision repair, interesting cross-link repair, basic excision repair, homologous recombination repair, mismatch repair, and non-homologous end joining. Our hypothesis was then that maybe these DREAM mutants could be resistant to any type of DNA damage. And this is indeed what we observed. Same assay here with ionizing radiation, significant resistance of the DREAM mutant Lin52, in this case, critically dependent on non-homologous end joining. We eliminate the repair mechanism. We have no effect. It's really via the repair mechanisms. Isolating lesions, completely different lesion type. Also here we see significant resistance of the DREAM mutants, and even single repair mutants that are hypersensitive can be suppressed by a DREAM mutation because other repair mechanisms can take over. Cisplatin, a highly toxic DNA damaging agent used in chemotherapy, for example, also here significant resistant to DNA damage. Then an really interesting observation here. We use here, instead of the post-mitotic cells, an early embryo undergoing the most rapid cell divisions of somatic cells in this animal to form an L1 larvae. When we apply ionizing radiation, these embryos are dead. You see this in the solid bars here, that with increasing IR, the embryos die. A DREAM mutant is sufficient to revert this and mediate resistance even in proliferating cell types. And what is more is that the BRCA mutant, completely deficient in homologous recombination repair, similar to BRCA carriers in the human population that have a very high cancer risk. Here we can reinstate resistance to DNA damage because other double-strand break repair mechanisms are now hyperactive. They work better. Now you might argue, I'm not a worm, I'm a human. Is that helping me in any way? And the DREAM complex is highly conserved. It's the exact same components of this repressor complex that are present in humans as are present in C. elegans. So we ask whether among the DREAM targets that are known to be directly bound on their promoter sites in humans, are there DNA repair genes enriched among them? And we find a significant enrichment of DNA repair genes, indicating that the same genes, repair genes that are repressed in C. elegans by DREAM, could be repressed by the DREAM complex in us humans. In humans, some things are more complex, such as that different subunits might operate also in other complexes. But in humans, we know of a kinase, the DYRK1A kinase, that phosphorylates a specific site on Lin52 that is required for the assembly of DREAM. We reasoned that if we now inhibit DYRK1A, either with a very well-established DYRK1A inhibitor, the natural compound harmine, or a chemical INDI as independent validation, we would inhibit the phosphorylation of Lin52, therefore leading to disassembly and inhibition of DREAM and alleviation of its repression of DNA repair genes. This could be a strategy to reduce cancer risk by reducing mutation rates and promote longevity by maintaining genome stability through better DNA repair. Does this work? So first of all, do we actually, with harmine and INDI, inhibit DREAM? So we here look at the expression after harmine or INDI treatment in quiescent human cells and find that many DREAM targets are induced, indicating that we successfully inhibit DREAM, and many of those are DNA repair genes. This led us to suggest that maybe we can now elicit the same resistance to DNA damage as in C. elegans, and this is indeed the case. Here we use UV-induced lesions, alkylating lesions to completely different DNA damage types that induce a strong apoptotic response in these quiescent human cells, and that can be reverted by either harmine or INDI, indicating that this chemical inhibition is sufficient to lead to DNA damage resistance. Can we use this in an in vivo situation? Our proof of concept phenotype was a degeneration of the retina, the outer nuclear layer that in Cockayne syndrome patients, I showed you this prodroid patient, lead to loss of the photoreceptors, and that is recapitulated here in a mouse model for the transcription-coupled repair, a DNA repair mutant CSB. And then we ask whether this reduction of DNA repair could be also driving the age-related macular degeneration in our population. And here we looked at transcriptome data of retinas from human age-related macular degeneration patients with age-matched controls, and see that the repair mechanism, the transcription-coupled nucleotide excision repair mechanism, the same that is induced by CSB is failed here, is downregulated in this human AMD, suggesting, giving us a rationale that if we now, by DREAM inhibition, boost DNA repair gene expression, we could bring the expression back and protect the retina. So we did this together with George Garinas, and Georgina and Kalina did this experiment on a prodroid mouse model ESCC1, where in the outer nuclear layer, already very early within 2 weeks of their lives, they show death here by tunnel staining of the photoreceptors, and it is sufficient to treat them with harmine, with the DYRK1A inhibitor, to prevent the apoptosis of these cells. Do we prevent the buildup of endogenous DNA damage? Here in ESCC1, we see early in the life, in the retina, buildup of DNA damage stained by gamma H2AX. This is prevented by harmine treatment, indicating that this works in vivo. Then we ask, together with Emma Tailing, whether the DREAM inhibition could be a naturally evolved mechanism to extend longevity in mammals. And for that, we turn to the super ages, to the bats that we will hear from Emma later, much more in detail. where bats live way much longer than expected on their body weight. And we looked at the expression of genes that are induced in bats during their aging process, but not in human and mice. And we find that DREAM target pathways are induced, suggesting that bats limit the activity of DREAM to induce DNA repair and extend mammalian longevity. So what I showed you here is that DREAM mutants in C. elegans elevate resistance to DNA damage by improving repair kinetics. DREAM mutants de-repress somatic expression of DNA repair genes and confer germline-like repair capacities. In fact, all these DREAM targets are normally only expressed in germ cells. The DREAM complex inhibition in quiescent human cells elevates the resistance to distinct damage types. We can use the DREAM inhibition by hormone in vivo as a proof of concept that protects from retinal degeneration in the progeroid model. And we propose that DREAM inhibition offers the first pharmacological route for enhanced genome maintenance to prevent DNA damage-driven cancer development and aging. Now, for the second part, I want to really switch gears and ask about what are the prerequisites to transfer our results from basic research into clinical practice for anti-aging medicine. And there are two prerequisites for that. One is that we have biomarkers for biological age to predict anti-aging efficacy. We cannot do lifespan curves like we do in C. elegans with humans, impossible. And then, of course, we need preventive treatments that target causes of aging-associated disease and functional decline. We propose here that DREAM could be such a really novel type of such a treatment. Now, the pace of aging is very different in us humans. The biological age is different from the chronological age in most individuals. And this could be made measurable by aging clocks that really have been pioneered by Steve Horvath. And what most of the clocks do is that they look at methylation sites that are very precisely measurable because they are either demethylated or methylated. So it's like a 0-1 decision that makes them very quantifiable, and you can derive that, and then among all these 28 million sites of CPG potential methylations in the human genome, you can find some that correlate very well with the chronological age as that changes over the lifespan. So the aging clocks have really made biological age measurable because the biological age biomarkers are really essential to determine intervention efficacies. And these aging clocks have provided quantifiable biomarkers for chronological, and increasingly they can also be used for biological age. And it is very important to also apply these aging biomarkers for discovery science. The exact the accuracy of aging clocks has reinvigorated the debate of whether aging is programmed. And also we have developed a very accurate transcriptomic clock that actually finds then the underlying systemic pathways that are enriched in this aging clock. And their epigenetic clocks also show enrichment for developmental sites. And that was kind of suggested by some people as signs of a kind of program. Now, on the other side, however, there's entropy-driven aging. And many studies indeed show an increased variation, noise with age, or show stochastic processes such as DNA damage driving the aging process. So how could that be resolved? So is really the accuracy of aging clocks compatible with the evolutionary theory of aging that I alluded at the very beginning, based on the Weissman concepts? So DNA methylation data are highly correlated with chronological age. The biological pace of aging can be derived from the deviations from chronological age, and thus its accuracy suggests a programmed process, while the evolutionary theory actually predicts that aging is caused by the vanishing selective pressure on somatic maintenance once the genes have been passed on to the next generation. How can we reconcile this? And the way we approach this, together with David Meyer, who did all the bioinformatics here, is that we ask whether we can generate clocks based on purely simulated data. So without any biological input, just purely theoretically. And the approach we took is that we defined a ground state. The ground state means the age that you were at the beginning of your individual life. For example, after the fertilization of the egg, the zygote that you all grew out from. So there must be an age 0. And what we did then is we defined a ground state by just making out numbers, 2,000 numbers between 0 and 1, that we defined as a ground state, completely randomly chosen numbers. And then we added for each year 1 round of stochastic variation. And so at the age of 1, there would be 1 round added. At the age of 2, there would be 2 rounds of stochastic variations on each value added. And at the age of 100, 100 times purely stochastic variation would be added. And we defined 6 data sets, each one comprising of these 2,000 numbers, that we trained. And 3 of them we trained, and 3 of them we used as independent validation. And each one is per each year a round of stochastic variation. And then we ask whether the simulated age could predict the age. And when we do this, we get no predictor whatsoever. It's absolutely flat, because stochastic variation, when it goes in any direction, of course cancels each other out if it can go into the negative, which of course in biology never happens. So we restricted then the output of the variation between 0 and 1, which is biologically meaningful because every value you measure in biology can be normalized to values between 0 and 1. And then we get a perfect, completely simulated age predictor with an extraordinary high accuracy. So it works. How many features do we need? I told you we made up these 2,000 numbers. And with 2,000 of these features, of these numbers that are randomly simulated, we get a very accurate age predictor. It's enough to use half of those, and this is a number that in omics is relatively small. And that is enough without any biological knowledge to generate a clock. What drives the clock? So here it is the amount of stochasticity we add in every time step. Here's our original 2 standard deviations. When we halve the amount of variation, stochastic variation that we allow for each round, we slow the predicted age. If we double the amount of stochastic variation we add in each step, we accelerate aging, suggesting that it is the rate at which stochasticity accumulates in aging that drives the aging clock. This also works for transcriptome data. Here we use real biological data as the first step to define a ground state, and then ask whether a stimulated age could predict a prediction we have generated with an accurate, with a highly accurate transcriptome clock, and we can significantly predict the age. Here we use 1 biological sample to then ask whether we can actually, with a predicted age based on pure stochasticity, we can predict the biological age. This is the elegance where we know the biological age. Very different to humans and mice. We know the biological age. We can predict it. Can we predict accelerator and decelerators that are known to change lifespan? And we can dissect here by purely stochastic data-based clocks the age acceleration of short-lived animals and the deceleration of long-lived animals. So it works in real life. Then we ask, because the most state-of-the-art clocks that are used nowadays use single-cell methylation data, CpG methylation data. We simulated that by defining a ground state for single cells, where we used, for each simulated methylation sites, we used a value that is an average as a ground state of 1,000 single cells that we purely simulate. It's a pure simulated experiment. And then for each of these sites, each methylation site can be 0 or it can be 1. And the average, for example, 0.13 means that among the 1,000 cells, 130 sites are methylated, 870 are unmethylated. And then for each round of stochastic variation, we defined maintenance efficiencies and de novo methylation efficiencies. So it's how much maintenance is experienced by every site and how much de novo methylation makes up the variation of stochasticity that we are adding. And we do this again for all these cells and then average it and use another round of stochastic variation. The maintenance rates of 99.9% are giving us a very highly accurate age predictor. These are now completely determined, artificially determined maintenance rates that are sufficient for this, and it gives us a very highly accurate age predictor. How many features, how many methylation sites we need? It's enough to have 32 simulated CpG sites based on purely stochastic accumulation to generate a very precise age predictor. What is driving the pace of predicted age? Again, it's the degree of stochastic variation, which for methylation sites is determined by the maintenance efficiency of each methylation site. So the higher the maintenance efficiency, the slower the predicted age will advance. The lower the maintenance, so the more variation can there be for demethylations, and the lower the maintenance rate, the more accelerated the predicted age is. And then we ask whether we can indeed predict the age of biological samples by just using a ground state of the youngest biological sample and just purely here in this case with a fixed maintenance rate, we can predict the age of the sample just by adding stochastic variation. We can indeed predict the Horvath clocks very accurately with estimated maintenance efficiencies, where we just estimated from biological data. But we don't even need to do that. We can use a universal maintenance efficiency, so completely simulated data, to predict the Horvath clock. We can do that also with second-generation clocks, the pheno age. We can have accurate predictions purely on simulated data. This also works for real-life samples. Here we use almost 400 healthy human blood samples and define the only biological information we use as input is the methylation pattern of the youngest blood sample. Everything else from then on is simulated and can predict the chronological age of these human blood samples. We can expand that further here to over 11,000 independent samples from 15 independent data sets. We only use one single sample to define a ground state. Everything else is stochastic variation, and we can the accumulation of this variation is sufficient to give us an age predictor of the chronological age of all these samples. Recently, Steve Horvath published a pan-mammalian clock that works across all mammalian species based on methylation sites. We asked whether a stochasticity, a purely stochastic-based clock, could also make an age predictor for all mammalian species. And this works. Here we use one single biological sample. One of the youngest in all these analyses is from a dolphin. And that is enough to make an age predictor across the mammalian kingdom. It doesn't matter whether we use this dolphin sample or any other young sample from any other species. All of them give us a very accurate age predictor only based on accumulation of stochastic variation. Can stochastic-based clocks, can they predict interventions? Indeed, they can. Here we use 3 different mouse models that have delayed aging, growth hormone knockout, TET3 mutants, or calorie restriction. And we built 4 different stochastic-based clocks, just with different maintenance efficiencies. That's only one difference. And they detect the deceleration of aging significantly. And they also, in humans, detect acceleration of aging here in a set of smoking humans. Parabiosis experiments where heterochronic parabiosis, a combination of a blood circulation of an old mouse with a young one, can decelerate aging. We can measure this here with a stochastic-based clock, that heterochronic parabiosis slows biological aging. And we can also detect the reprogramming, the resetting to a younger biological state by cellular reprogramming, induced pluripotent stem cells, starting out with a fibroblast that in the course of 28 days, we see that the stochastic variation-based clocks is reverted as a result of the reprogramming. So what I conclude here is that we can make aging clocks based on accumulating stochastic variation. The simulated stochastic variation provides an accurate chronological as well as biological age prediction. Any data type with a ground zero state at age 0 can provide an aging clock. So it does not matter what we're using for aging clocks. Anything will work that is set to a 0 stage at the beginning of an individual's life. The age-dependent changes in biological parameters are driven by increased stochastic variation. The degree of the stochastic variation determines the pace of aging. And we would predict that geoprotectors that could reduce the variability and restore the regulatory tightness could be most effective. And the accuracy of aging clocks is compatible with the evolutionary theory of aging, because what drives aging is just the lack of maintenance resulting in accumulation of stochastic variation in biological processes. And I acknowledge the people who did the work I showed you. I thank all the funding sources, and I thank you very much for your attention, and I'll be happy to take any questions. Thank you.
Thank you, Björn. And I will not thank you for having run out of time, because I bet there are a lot of questions and we only have time for one. Who's going to ask the question? Morten, in the front. Oh, okay, yeah, sure.
Yeah, thanks a lot. Regarding the first part, do you think if you are inhibiting the dream complex more and more, and then could you do it? Could you overdo it? Could you have too much DNA repair? Is there a U-curve in the end?
So we don't see any adverse effect of just having DNA repair. I think the limitation, why is it limited at all, DNA repair, is probably most critical in embryonic development, that you actually have rapid cell divisions where you have to compromise on repair. You cannot stop the whole embryonic development because you want to perfectly repair every region of the germline. So it makes sense in somatic cells to limit it. But in adulthood, it's probably far less critical what kind of replication rates you have, so that we believe this could actually be quite a safe method. But of course, this will be important then to determine what kind of dosing, for example, of the pharmacological compounds elicits the most beneficial effect.
Thanks, Björn. Great.